today. Today's lecture is an introduction to introductory ecology. We're going to learn a bit what is ecology, the definitions, learn a bit about scientific thinking, organizational levels in ecology, types of experiments, and a bit about the importance of ecological research. So, ecology. Ecology, a bit of etymology here, comes from two words, the study of, logos, like everything else, and oikos, which is Greek for home. So ecology is the study of ecological research. And this is our home. Economy, for example, is the management of our home. Ecology is the study of our home. And framed by this guy, Ernst Haeckel, who was like the first ecologist before even ecology happened. Ecology is a relatively recent discipline, recent study area. So Haeckel defined it first as the study of the relationship of organisms with their environment. Very specific, very simplistic, organisms and their environment. And then later we have a definition that is the known one today by Krebs, the scientific study of the interactions, biotic and abiotic, that determine the distribution and abundance of organisms. So again, I'm going to repeat that. We are looking at the interactions between organisms and their environment, and their environment can be biotic. What is biotic? Natural? Yeah, living, other organisms. Biotic, other organisms, so interactions of organisms with other organisms, or abiotic interactions. These are interactions between organisms and their not living environment, okay? Everything else that is around us that is not organisms, not biological entities, that determine the distribution and abundance of organisms. So for example, we have this beautiful tulip from the desert, from the Negev desert. It grows here, naturally in the Negev desert and only there. And we can ask, look at the distribution and abundance of this tulip. And we can ask, why is it growing in the desert? Where do we find it in the desert? And what are these interactions with the environment that determine when and where we find this tulip there? So what type of biotic interactions can you think about of this tulip? Other organisms? Anything? Sorry?
Insects.
Pollination? Sorry, my hearing is bad. Insects, sorry, yes. Insects. Insects can pollinate, but they can also what? Eat it, yes. So this can be positive interactions, this can be negative interactions, they can be herbivores that feed on the plant. What are the types of biotic interactions do you think the tulip can encounter? Animals that feed on it, animals that pollinate it. What else? What other type of biotic interactions? Fungi, yes, fungi that can either have a negative effect or maybe a positive effect, right? We know fungi that can help plants. Good. What else? What other types of interactions? Biotic. Please speak up. People. Yes, that is a type of biotic interactions, although that is that's like a completely other type of interaction, and these are like anthropogenic effects. We're gonna maybe try not to include those. Something that is natural.
Just want to say that not natural.
Not natural. Yes. Yes.
Biotic, like an abiotic, biotic interaction would be like rain and the effect of the.
That's abiotic, right?
Yes.
We'll get there. But more biotic with other organisms. Other plants, yes. What type of interactions will be there? In speaking Hebrew. Afraya, yes. So reproduction, good. Yes, they need their mates to reproduce, good. What other type of interaction with other plants? Yes, dominancy of other plants, competition with other plants, very important. Plants cannot move. So if they happen to be next to another, let's say a big tree here, it's going to be big competition. Okay, so these are interactions that determine, biotic interactions that might determine whether or not we will find a tulip in a specific place. If there are no neighbors to reproduce with, if there's a big tree to compete with, if there are no pollinators, if there are herbivores that feed on it, they will determine whether or not we will find this tulip there and how many of the tulip we will find there, the distribution. What type of abiotic? You had ideas about an abiotic? Yes. Rain can determine whether or not we'll find the species here. Like what with rain?
Oh. The area doesn't have enough rain.
Yeah.
But also maybe could affect underwater, but water that is underground water.
Mm-hmm. Okay, so reserves underground. Yes, I don't think tulips have deep enough roots for that, but rain is a very important factor, especially in the desert. So we maybe find this tulip in crevices, in the soil, or in areas where we can find more water there. Cool. What other abiotic factors? Yes. What does that mean? The water. Soil erosion, you mean? So you mean this affects the tulip or the tulip is affected by it? Okay. So, well, nah, not really, but okay. Has to do with the soil, right? The soil is an important abiotic factor. Yes. Okay, there's also wind. Sorry?
Soil quality.
Soil quality. Yes, soil quality is very important. Sunlight, yes, for plants, especially, very important. Temperature, yes. Wind, of course. All these factors, yes. Floods, yes. If the water is, the stream is too harsh, definitely, yes. Cool, good. These are all important abiotic factors that will determine whether or not we will find this particular tulip in this area. And we talk about distribution and abundance, both in space and also in time. Ask, when do we see this tulip? Do we see when do we see these tulips?
Specific dates.
When-ish?
Winter.
Winter, right? Winter, spring, somewhere in between, right? And that is particular to this area, to the Mediterranean, to this arid environment, because we only have rain in winter, and we don't have rain in other seasons, which is unlike other places around the world, and we will find this tulip only there. Okay, so we discussed this idea of species abundance and how it is affected by biotic and abiotic factors. And back to this ecology. What is ecology? Ecology is mainly part of biology, part of biological studies. I studied my bachelor in biology, but it's not really biology. Is related to biology. In biology, we study evolution, we study physiology, we study genetics of organisms, but in ecology, we don't study that, but it's related to all these things. They're connected because it's part of the biological science, life sciences. But ecology is not only that, it's also heavily related to geography and geology, because we talked about the abiotic factors, right? And it's also related to sociobiology and behavior, because we also talk about the way organisms behave, how they interact with each other. So it's related to those. Well, so everything here is related to ecology. And when we talk about different questions, when that ecology seek to answer, again, talk about the where and when do we find species? Where do they occur? What is their abundance in the time and place that they occur? Do we find a lot of them? Not so much. And when and where? But the most important question is, why are organisms more abundant in that particular time and in that particular place? In other areas of study in biology, for example, we study biology, the genetics, we study how things operate. For example, in physiology, in plant physiology with the tulip, we will ask, how does the tulip fix sunlight, fix carbon and it creates sugar from sunlight energy, right? And how can it take water from the soil? What are the genes that are involved in that? In ecology, we relatively less ask these how questions. We ask the why question. Why is this happening? Why do we find the tulip there? Why is it abundant in this particular time, in this particular place? And for the international guys here, I want to teach you a word in Hebrew, and that's lama. This is why. We will keep on coming back to this question, the lama question. So my contribution to your Hebrew, lama. And that's the most important question in ecology. Keep on asking why. Okay, and now I'm gonna briefly go over this scientific thinking in ecology. This is actually not the scientific thinking in ecology. It's all disciplines, physics, chemistry. They all go through the same process. We start with an observation, science, and then we think about a hypothesis to explain this observation. And then that's like the why question, right? And then we go to the literature, look for what we know already, what can we learn from the literature on the topic, test our hypothesis with experiments. We analyze the data, and then we either support or reject the hypothesis. If we reject the hypothesis, we go back. We try to understand what was wrong about our hypothesis or about our experiment. But in ecology, this bit is very important, observation. How do we get an observation in ecology? How do we find an observation? How does that happen? Very simple. It's not a complicated. Watching what?
Nature.
Nature. We go outside. We go to nature. We observe. We look at things. We look at nature. And then we find patterns. So you can find patterns in space. You can find patterns, like I said before, in time. This is an example for a pattern that you can find in time. These are examples for two species that I hope many of you just saw. They're just now finishing flowering. These are what we call autumn indicators, מבשרי סתיו in Hebrew. These are plants, a group of plants that flower at autumn. They flower at the end of the dry season, at the end of summer. That's the sea daffodil, חבצלת החופש in Hebrew, חצב in Hebrew, the sea squeal, and you all know them, right? In them. And a pattern that we can see, what pattern do you see here? Let's say the pattern in time is pretty obvious. We see plants that flower at the wrong time. Everybody else is flowering in spring, right? Like the tulip we saw before. They flower in spring. Why does everyone flower in spring?
Rain.
After the rain, they got the rains. They have now water. Insects are in the air. They all come. There's pollination. It's a celebration after the rains. Why do these guys, that's the Lama question, why it's pattern in time, we find them flowering before the rainy season. They start to flower before the rainy season. And that's a pattern that we see in time, and then we ask why. Another thing that we can see in these plants is that they're very showy, very bright, white, very big, right? The sea daffodil is this big. I'm sure you've seen it on the Tel Aviv. There's loads of them everywhere on the promenade, on the sea, next to the coast. And the sea squeal is also very big, showy. Also a why question, why? Why do these plants that flower at the end of the dry season, when no one else is flowering, you can see everything else is dry all around them? Why? What is the answer? Less competition for pollinators, right? No one, there's pollinators, bees are there, and see that all the bees in the neighborhood, they come and go to these plants. They also smell really good. They're very attractive for all the pollinators around them. And there's no competition. So it's a good strategy to be the only ones, and having these showy, big flowers, you can come and then be pollinated. And these particular species, they have storage below ground, so they can use water, the reservoirs from the water of last year, have them below ground in these big bulbs, like onions, and they can use them to flower before they make leaves. They don't have leaves. These are all dry leaves from last year. They first flower, and only then, when the rain comes, they produce leaves for next year. Okay, so that's a pattern. That's an example for a pattern that we can look for before we start formulating a hypothesis. Any questions up to here? So where do we look for patterns in ecology? We have several organizational levels. We have what we call an individual level, where we look not just at single individuals, but it's an individual level. So we look at questions like the responses of organisms to their environment. We're going to look at examples. We have the population level, and we will talk about that at length in the following weeks. But a population is a group of individuals that belong to the same species, that occur at the same time in the same place. What is a species? We'll also discuss that a week or two from now. But I think you all kind of have a vague concept of what a species is, right? We all belong to the same species. We are all Homo sapiens. And so we can be defined as a population of the Introduction to Ecology class. This is a population. We all occur at the same time, at the same place. We're a population. A community is when we have different species that occur at the same time in the same place. And an ecosystem is when we add to all these species also the abiotic factors. And I'm going to give you just some example to explain that. Just so you understand, in ecology, everything that we're going to see, and this is how we're going to learn this in this course, everything is built up in these, according to these organizational levels. So these are layers. And we start with evolution, which is more at the individual or the individual and population level. We'll talk about that next week. And then we move on to population ecology and talk about small populations. So we scale up to the population level. And then we move on to species interactions, positive, negative species interaction. We scale up to the community level because these are already interactions between species, not just within a population. We'll talk a bit about behavioral ecology, and then we move on to community ecology and then ecosystem ecology. So the way this course is built is also according to these organizational levels. Okay, so again, back to the first level, individual level. We look at responses, effects of those different abiotic and biotic factors. So not specifically one individual, but a group of individuals, but they do not interact with each other. We care only about how they respond to other factors. That's an example for a study at the individual level. This is a study that looked at sugar concentration or sugar quantity in this in the flowers of this plant, Thymus vulgaris, Coranit מקורקפת in Hebrew. I don't know if you know this plant. It's part of it's a spice. It's part of what is used for this the za'atar spice. I don't know, right? It's part of that. It's not only a spice, but it also is a very attractive plant for pollinators. And in its nectar, tzuf in Hebrew, it produces a lot of sugar. And in this study, they looked at the effect of drought, so less water, on the production of nectar, on the content of sugar in this nectar. See, clearly that there's a reduction in the quantity of sugar in under drought conditions. And then, of course, we ask, Lama? Lama? Less water, less energy, right? Less energy to produce everything, right? So there's also less energy to produce sugar and served for other properties in the plant. Okay, so that was the individual level. Population level, again, population is a group of individuals that belong to the same species that occur at the same time, the same place. Here, we ask a lot of questions that are related to demography. We look at demography, population growth, population decline, which factors affect the growth or the decline of population, how many individuals are added to the population each year. Is it growing? Is it in a steady state? What type of factors affect the demography of populations? Do populations from different areas differ in their demography? Is this population about to become extinct? Is it growing? That's a lot of demography, similar to demography that we have for humans. A lot of that is this. That's an example for a study at the population level, where they looked at the density of these sparrows in California over time. So you look at basically demography of this population. You see how many of them you can find every year and what happens to them. See that there's a decline over the years in the density of the sparrows, right? And of course, you can now try to correlate that with other factors. Maybe things are getting warmer, maybe they lose their habitat. We'll talk about all those things later during the semester. But that's an example for what we do when we talk about populations. Questions up to here? So we move up the scale from population to the community, and now it's not only one population, one species, we have several species that occur together at the same time in the same place, and their interactions between them. So in this case, we ask about all these types of interactions. Before, it could be predation, it could be pollination, it could be mutualistic interactions, it can be also competition between species. And we also asked questions that are related to the idea of species diversity, species richness, how many species occur in a particular time, in a particular space, how is this affected by different conditions. Species diversity, we're going to get into that, is a particular calculation that we do when we try to take into account not only how many species are found in a particular area, but their relative abundance. So I'm sure you've heard this term, species diversity, biodiversity. This is the community level type of question. That's an example for that. This is a study that looked at diversity. So how many species of butterflies and the relative abundance occur in Sweden and Finland in this case, and how it changed over the years, 120 years, roughly. And also another thing that changed during this time is climate, and you can see that there's warming area 1901, and this is 2019, and you can see that there's an increase in temperature, less blue, more yellow, more red. With time, we know climate change occurring, and what they wanted to look at is species diversity or species richness. In this case, it's the number of species that occur in these countries and how it is related to temperature increase. What can you see here? Unlike maybe what you would have expected. Yes, there's an increase in diversity with temperature, with the rise of temperature. So good news, right? This is good. More species with the climate change. Why do you think we have more species with climate change? Predation. Maybe less predators, but I mean, why would there be less predators? Because they're affected the opposite way by maybe. But migration can be a very good explanation because we're talking about maybe the northern limits of these species cannot go beyond that. So what can go on here is that species that used to live here now migrate up here because this is too warm for them, right? So up here in Scandinavia, we have enrichment of the species, more butterflies, this is fun, but probably what is going on here is that there's a decline because they migrate upwards. So maybe good news for the northern countries, southern ones. Okay, so that was the community level, and now we scale even higher up at the organizational level, and we move on to the ecosystem level, and we enter the abiotic factors into our species that coexist at the same time and the same place. And here we ask questions that are related to energy flow in the system. We talk about food webs and how they interact and how the energy flows from the primary producers that are the plants to the herbivores that feed on them, to the predators that feed on them, and so on and so forth. questions that are related to these ideas. So, for example, we can talk about energy flow. We can talk about also carbon flow, nitrogen flow, etc. So, energy flow in the ecosystem. Energy is can be used mainly by plants. Plants are among the only organisms that can fix carbon and use the energy from the sun to do that. And so they are the primary producers. They produce the first sugars, and then they are used by herbivores. So the energy moves from the plants to the herbivores, and from the herbivores to the carnivores or the omnivores that eat everything. And then it moves to the decomposers and the nutridivores and the microbes that recycle everything and take it back into the energy cycle or leave it in the ground somewhere. So that's an example for what we ask, the type of questions that we ask at the ecosystem level. Questions up to here? Yes.
What the number of species? Doesn't put the number of individuals.
Wait, where the butterfly?
Yeah.
Here?
Those who used to live with maybe number of individuals. I mean, it's graph is about the number of species, not
Yes,
species.
yes, you're right. We talk about species richness is number of species. That is a very, you're right, that's a very crude estimation of actually what's going on there. This is why, and we'll talk about that when we talk about community ecology, this is why we often use diversity and not species richness, and diversity takes into account the relative abundance of each species, because this can be made of many individuals where there's many species where there's one individual left, let's say, and that's obviously horrible, right? And when we look at diversity, we look into that. You're right, yes. Any other question related to that or to the ecosystem? Okay, so we had an observation, looked at nature, we formulated a hypothesis, we looked in the literature to understand what we know so far, and now we move on to test this hypothesis. And I'm going to show you a few examples for how we test hypothesis in ecology. What types of experiments do we do? And in ecology, we have the basic two types of experiments that we have, and that's just an extra one that we'll talk about. But mainly we have observational experiments and manipulative experiments, real experiments, controlled experiments. So we're going to talk about these. Observational experiments are experiments where we do not manipulate anything. We go to nature and we observe without having a controlled treatments, not touching anything. We look at it as is, and we want to find out, for example, species richness, species diversity, which species we find where, or we can also talk about population level. We want to see how population changes with time. We just observe. We do not touch. So these observational experiments, because nature is chaotic and there's a lot of variation in nature. It looks nothing like a lab. There's variation. There are many factors that affect what we see in nature. So these observations need to be very systematic, and they need to be done with enough samples so we know that we get closer to reality. When we sample, we cannot sample very few samples. Because there's just a chance that when we sample these particular areas, just by chance, they're very different from what is the mean. So in order to get closer to reality, to the mean, we need to do a lot of sampling, and they need to be systematic. So and they can take place in different scales. So for example, at a very local scale, I can ask, what is the diversity of insects along rivers in Europe? And then for that, I need to go to, not enough to go to one river, I need to go to several rivers, and I need to maybe be randomly sampled. And when I go to these rivers, it's not enough that I go to that particular area. I need to go throughout maybe few meters from the river on both sides, because that particular side can be different from due to different factors from that side, and it's not enough to sample here. I also need to go downriver or upriver, so I need to do a lot of sampling, and they need to be systematic, and also random. So that's an example at a very local scale, but I can also sample species diversity, let's say insect diversity. I can see, I want to know if it changes along altitudinal gradients, if there's a difference between the top of the mountain and the bottom of the mountain in, again, let's say insect diversity. So I need to sample heights and at different mountains. And again, it has to be random and static. Or I can go scale up and do at a regional scale, and look at throughout Israel, for example, look at species diversity and see differences between the desert and the Mesic Mediterranean. Or I can even go global and look at species diversity throughout the globe. That is really hard to do. You need a lot of collaborators, or maybe do a meta-analysis. We'll talk about that again. That's an example for a pattern that we see at the global scale. So this is not going to nature, but you need to zoom out and see a pattern here. And the patterning that we see, that we often find in different organisms, is that we see more species around the equator. Species richness, species diversity is usually, for most organisms, higher near the equator. Then I can ask, Lama? What would be the answer? We'll have a whole class almost dedicated to that, answering that question, but just maybe someone want to throw
Sun energy.
Sun energy higher at the equator. Why? Equator, right? It's a neat. The sun is in closer, and there's more sun energy, so maybe that. But there are other reasons, other hypotheses, and we'll talk about that a lot. Okay, so how do we do that? We said we needed to do it in a systematic way. If you're sampling organisms that do not move, like plants, use these sampling quadrats, and you go and you sample. You spend hours upon hours of sampling. You place them somewhere in the field, and then you sit for hours and count all the individuals that you see there, all the species you see there. Count them, and you write down the names and the numbers of everything you find there. And you can do that also with corals or all species that do not move. That's fairly easy. Can have all kinds of ways to create quadrats. And we will talk about, when we talk about community ecology, we'll talk about what is the size of a quadrat, because there's a whole theory of what is the size of a quadrat that is and relevant for particular species. We'll talk about that.
A quadrat is useful for figuring out the density of organisms in a certain area. I often use quadrats that have strings across them in both directions. And what I use that for is to look at all the points where the strings intersect and I observe what is under those intersections at each point. So I'm looking at what's under all the points of intersection as like a random way of figuring out what organisms are covering the area. Also typically count all the mobile organisms, that's all the things like snails, those just get counted.
Okay, just another example for how to use a quadrat in ecology, not for plants. You can also use transects. Transects are these lines. You determine the length of these lines in advance. You need to understand what is the representative length of a transect of this line that you go through, and everything the line touches, you count. Instead of a square, a quadrat, like before, you have a transect. Everything that touches the transect is accounted for. You count it, or you write down the species name and how many, right? That's another way of doing that. We don't have any highly sophisticated ways of doing that. That's at the basics. When you need to do that, this is two. For insects, for example, or for rodents that move around, we use what we call pitfall traps, which are traps that we have in the soil. And then if these insects do not fly, they fall there, and usually they are along a transect. So you have few along a transect, and you need to go there, pick the insects before anything happens to them. The minute they fall, you need to pick them out, measure them. I don't know what this study did, weigh them, count them, identify the species, put them back again, or mark them so that next time you catch them, you know that it's the same individual or not. There are pitfall traps for bigger animals. We cannot use pitfall traps. Like for mammals, you use camera traps. These are really good if the animals have markings that are individual, like in leopards. You can identify a particular individual according to the pattern, the coat pattern of these leopards. This is a neat observational study that look at seals, and they use facial recognition. Facial recognition software was designed for humans, for facial recognition, for security. And they use it for the seals, and this way they identify individuals in pictures, and they can look at the demography. Where do we find these individuals? Where do they move to? Who they mate with? That's another way of observing. Can use citizen science for observations. This is Penguin Watch, where you can enter there and help be part of this experiment and help count penguins. They give you a picture of penguins from somewhere in Antarctica, and then you can count how many individuals you see there, and this goes into study. They have cameras in various areas and take pictures all the time, and then you can help as part of the citizen science. This is how you find, this is how you do observational experiments in the field. Questions up to here? Okay, so these were observational experiments, but the real experiments that we do in ecology are what manipulative experiments. These are controlled experiments with controlled treatments. So you not only go and observe what is there as is in nature, you manipulate it. And in these experiments can be from a very tiny scale, very accurate scale. These can be lab experiments, but these can also be field experiments. As long as there's some manipulation, they are not pure observational experiments, they are manipulative experiments. So again, they can be at different scales, different precision levels. So for example, this is a study that looked at behavior of ants from different colonies, and they looked at aggression of individuals or at the brood care, the care for the young. So here they had dead ant from a rival colony, a different colony, and they wanted to look at the aggressiveness of the workers. And you see, this one is very aggressive. This one doesn't care, not very aggressive, doesn't even look at this other individual. This one really is aggressive. This one somewhere in between. And here they looked at brood care. So they had this baby ant, a pupa, and they look how they quantified how many times it touches with its tentacles the pupa to care for it. So this one you see takes care of it very gently. This one doesn't care, walks all over it. So in this way, they wanted to see if ants from different populations behave differently. But this is super controlled. Petri dishes under artificial lights with cameras on them, and there's a software that quantifies the movement. This is not how it looks in nature. Completely artificial, right? But also very accurate. What's the advantage of doing that and not going to nature? More controlled. Things can happen in nature that are not here controlled for all other factors. You eliminate all other factors. The only factors you care for are the ones you see here. But it's also not natural. It's not really how these ants behave in nature, maybe. So that's the downside of doing these controlled experiments. That's an experiment from my lab. We look at competition between plants. So this is not as artificial as the Petri dishes, but this is also fairly artificial. We have plants growing together. We have them in these transparent pots, so we can look at the roots, how the roots grow, and take a picture and see the movement of the roots and how they grow from, how they differ between plants, if they have competition with different types of plants, different species. So again, less controlled, but also nothing like it looks in nature. In nature, these plants don't grow in these plastic pots. And this is how a field experiment, more natural experiment looks like, but this is completely controlled. What this experiment wanted to ask is, what is the effect of competition on the germination of seeds? So this is an area next to Sde Boker. What was done in this experiment is that there were different quadrats. In this quadrat, they removed all the vegetation except for, and they had seeds of this particular species here. Adjacent to that, in this quadrat, you cannot see it very well, but they also had these the same seeds, and they wanted to see where do they see the seeds grow better, here or here. And you can see repetitions, this pair and this pair and this pair and here and there. And again, this is less artificial, this is more natural, but what can be problematic about this? Said before, other factors, right? Cannot control for all, which factors you cannot control for in this case? Well, light is...
But animals eating,
Animals eating, maybe there's an animal that really likes to feed on the seedlings here, but not here. And then at the end, we will think that this treatment was bad, so competition is bad for the plants, but it's not the competition, it's the animal that comes to eat there. Right? So there are other factors you cannot control for. which makes things complicated. But on the other hand, it's closer to nature. This is how it really looks like in nature. So there's a trade-off. By the way, what do you think was the effect here? Which seedlings were happier with competition or without competition? It's plants that grow here naturally. Sorry? Without competition? I'm going to tell you that they were happier with the competition because this is a desert area. And if there's no competition, it's completely exposed to the sun. It gets very dry very soon. They all die. Okay, this is another example for an experiment in the field, but also completely controlled. This is an experiment that was done in Mashabim sands, also down south, where they looked at the risk taking of these gerbils. How much risk will they be willing to take if they place these seed trays in different distances from the bushes? Their burrows are next to the bushes, so they live here, and they are active at night, like many desert animals. They go out at night and they look for seeds. So they place these seed trays at different locations, and they wanted to see if they will take the risk of going out into the exposed area. And what they did, not only they had these seed trays, which is artificial, they also had a trained barn owl, tin shemet in Hebrew. It was trained. It did not prey on the gerbils. It did not try to attack them, but it flew. There was a trainer standing, and the barn owl flew across the field, back and forth, back to the trainer. But just hearing this flight and the cries of the barn owl can be scary for the gerbils because they naturally occur there. But again, this barn owl was trained, so it's a controlled experiment. But that's another example for how to have a controlled experiment in the field. Questions up to here? Okay. So these are the main two types of experiments, but we also have what we call natural experiments. If there's some natural phenomenon that happens, it can help us to compare what is going on when this thing happened. And when I show you the examples, you will understand. So, for example, when there's a new lava bed that is formed, and now go to this area which is completely pristine. It's just rock. There's nothing growing on there. We can see now the successional stages, which plants come first, which organisms appear first, start to grow. How long does it take for this to turn into a forest? So we can use that as a natural experiment. That's another not so natural experiment. That's an example for an oil spill. It's a horrible nature disaster that happened here in Israel. But a lot of scientists were busy after this happened, looking at what happened to the organisms there. How do they deal with this? How long does it take for this nature reserve to, hasn't yet, but to go back to its functioning state, let's say? Which species are more affected by this oil spill? How far from the oil spill is there an effect? And these are things that happen by mistake or randomly, but we can use them as natural experiments. What's the problem with these natural experiments? That Okay, time. Let's say we have a lot of time to follow that. But what is another problem with that? We said before, sampling. We need a lot of sampling. We don't have the before and after. We don't have a control, right? What is the control for this? What do you compare this for? Yes. Yeah, because this is one occurrence, right? One time, just one. What do you compare it to? There's nothing to compare this to. This is one repetition, one point in time and in space, and you cannot repeat it. You cannot compare it to anything. Hopefully we will not have another comparison to this, right? It's not that we want this, but so it's kind of a tricky experiment, but we still use them sometimes. Okay, some quiz for you. This is an example for studies that simulate climate change. This is the use of rainout shelters in the Swiss Alps, where they shelter different plots from the rain. So there's less rain here. This simulates climate change because we'll have less rain compared to these control plots here. That's an example for an experiment in Finland when they wanted to look at warming temperatures. So they have these tents here. They don't shelter from the rain because they're open, but they keep the environment warmer inside. So they want to see the effect of a higher temperature compared to this control plot on the plants there. You can see these reactions here. Are these observational, manipulative, or natural experiments? What are they? Manipulative, of course, because you manipulate stuff, right? Even if we look at species richness, this is still manipulative. This is an experiment with this invasive plants, invasive plant, an anemone, nurit in Hebrew. In Israel, by the way, it's a super rare species. You will find it only like in super protected areas up north in Pula, super protected and rare. But in North America, it has become invasive. And what they did in this experiment is they looked at areas with the invasive plant and areas without the invasive plant, and they looked at species richness. So how many species are here? All of this is this anemone, this invasive plant, and this is where it's absent to see the difference in species richness. Is an observational, manipulative, or natural experiment? What do you think? Observational? Yes, definitely an observational experiment, but also natural, right? Because there's a use of this invasion from this plant. The researchers did not bring this plant there. This plant invaded due to other reasons in this area. It's an invasive plant, but it's being used as a natural experiment by the researchers to look for its effect. Okay, was not manipulated. The invasion was not manipulated by the researchers, but they looked at it. Any questions up to here about these types of experiments?
Observational experiments, but they find a certain anomaly in
Yes.
an existing.
Yeah, we can also use them for maybe more controlled experiments, but yes, they use either an anomaly or a natural phenomenon that doesn't happen very often, like the lava fields. Any other questions? Okay. And the last thing we have, in addition to experiments, are models. models are very important in ecology because we can use them to predict. If we want to look at populations or communities, we want to look at them in time or under different conditions, we don't have enough manpower or enough years to go over, look at a particular plant for 10 years. This is more than a single PhD. So you use models. You have a lot of data. They're data driven, so very hungry for data. But if you have the data, you can feed them to the models, and then at the end, you can predict, extrapolate what will happen to a particular population, particular community with time, given conditions that you add in the model. So we use them a lot. We'll talk about that too. An example for population growth or relationship between productivity and species richness or species diversity. We'll talk about that. Very good for predictions. That's just a simulation model that looked at the effect of the number of species in a forest, whether it's a monoculture or polyculture, how it affects the interactions or the end productivity of a forest. So instead of growing a forest for tens or hundreds of years, you do a model. You get all the data from existing forests, you feed it to the model, and then at the end you can learn about species interactions or the productivity of the forest at the end, given different conditions, different species that occur there. If you know the conditions each of these species requires from experiments. Okay, so when we look at ecology, there's this triangle, which is basically a triple trade-off between the different things we aspire for. And we aspire for realism. We want to know what really is out there in nature. But at the same time, we want to be precise, and we cannot be precise and real at the same time because we discussed this. If you go to nature, you cannot be precise. You cannot rule out other factors that are going on there. But we also aspire for generalization. We want to know not only what happens in a particular space in a particular time. We want to generalize. We want to know what happens in bigger scales. And we can use models for that or meta-analysis for that. But we cannot do all of them together. If we want to be precise, we cannot generalize. We want to be real, we can also not generalize. But that's like the tension that we have between these three ends of the triangle. We want to have them both. And when we do experiments, we try to take into account all of those. Okay, so the last bit is this data analysis, and I told you already that nature is very noisy, has a lot of factors that can affect. Everything happens very randomly. It's very chaotic, and this is why in ecology we are driven by data analysis. We do a lot of statistics, we do a lot of data analysis. We're not going to go into that in this class. I just want to let you know that most of the time, if we're not in the field, this is what we do. We do data analysis. And just an example from here, when I showed you this example for this thyme plant, Coronite, we looked at mean differences between the control and the drought, but you see that there's a lot of variation. Some plants have this amount of sugar in their nectar, some have this amount of sugar in their nectar. There's a lot of variation, and which is why we need statistics to deal with this noise and variation. Again, we're not going to go into that. Questions up to here? So, just a few minutes about why is it important to study ecology. I don't think I need to convince you that it's important to study ecology because you're part of the Porter School. But ecology seeks to understand nature in its complexity, but it's not only that. We said that it's a study of our home, and we need safe and functioning. And this is not news to you, but we are in the midst of a biodiversity crisis. I'm sure you heard about this. We have species disappearing all the time due to anthropogenic effects, mainly. What are these anthropogenic effects? Maybe very quickly. Main ones. Sorry? Climate change. Yes. Good. What else? Yeah. So, wait, but humans taking over the area, you mean? Yeah, so habitat destruction, right? Yes, good. Number one, by the way, habitat destruction, number one. Climate change, 2 or 3. Yes. Natural disasters, where does this fall? Mainly under climate change, usually nowadays, no? Climate change is both warming and these storms and fires, right? Good. What else? Yes. Disease. Well, yes. Yes. Well, it mainly affects us, but yes. Disease, yes. What else? Hunting, yes. Very good. Hunting. Over exploitation of natural populations, yes. What else? Pollution, right? Pollution, all types. And yes. Moving of species. What do we call that? The movement of a species? Nope. Yeah, well, yes, but on? Yes. Invasive species. Invasive species, by the way, are fighting over the second place with the climate change. Very, very harmful effect. Yes. Livestock, yes. Well, where does this fall? Part of habitat destruction, I think. The second place because they take over areas that are supposed to be for nature. Anyway, it goes on and on, the list. And all of these things, they lead to what we call now the biodiversity crisis. We're losing species. We're losing not only the animals that are very showy, right? Not only these animals, but mostly we're losing the tiny insects that no one cares about, and we lose them a lot. They're most affected, but no one knows, no one cares. Not only these guys. We have some endangered species in Israel. We have a lot of those. We will discuss them a bit, and we're getting to conservation a bit with these. Sure, you know many of them here, but we have some success stories. We have some. Due to all these studies, we study population, we study the conditions that can support population. We will talk about that too. There are some success stories and manage to save species and reintroduce species. We'll talk about that. Some examples also from Israel. Okay. Questions? Okay, so this was today's lecture. We talked about definitions, scientific thinking, organizational levels in ecology, types of experiments, and a bit about the importance. And you have an assignment for next week. And the assignment for next week is to look for patterns. And what I want you to do is take a picture, maybe from a hike you did somewhere. It can be also urban nature. It doesn't have to be nature in nature. But I want you to look for a pattern. This is an example that I always give. It's a picture that I took in German Alps, and what you can see here is that on one side of this lake, there's these trees growing, and they're not growing on this side. Ask Lama. Right? What do you think? Any explanations? Sorry. Yes.
What?
Sunlight. Yes. Same direction, right? Yes, that's a usual answer for things like this. It can be which direction the slope is facing. North facing slope, south facing slope gets more sunlight, less sunlight. In this case, if they get more sunlight, lush. In Israel, in the desert, if they get more sunlight, they have less vegetation in them. So yeah, that's a hypothesis. And what I want you to do is take a picture from your travels, from somewhere, write down where the photo was taken, look for a pattern, formulate a why question, like I did here, Lama. It's all there. It's all in the Moodle. It's all on the site. You don't have to take a picture of this. It's all there. And then what I want you to do is suggest a hypothesis, like we did today, explain why, explain Lama, and then suggest an observational experiment, one observational experiment, and one manipulative experiment. You can write this either in English or in Hebrew. And please do not have pictures or questions or hypotheses that are similar to the ones we discussed, including this one. Okay, not another picture of south-facing slope with more or less trees in it. And nothing that we discussed here in class. Yes. Question? No. You have a week to submit them in Moodle. Yes, please. Organisms, all types. Fungi, insects, mammals, plants, whatever. It has to be an organism, though. Yes. Yes, that's what I said. The lecture is recorded. It takes about a couple of hours after the lecture for it to be downloaded. So sometime later today. Yes, but the PDFs are already there. The PDF for this lecture is already there. Every week I'll update those. Yeah, you have a week to submit this. Any other questions? Yes, if you have a picture of microorganisms, it has to come with a picture of a pattern that you observe in nature. You can see microorganisms, cool. But I need to be able to see. Or Noam, the TA, needs to see it. Any other questions? Okay, so next week we start our journey with learning what is evolution and why is it so important for ecology. So see you